that everyone uh, who
Okay, recording
watches.
start.
Okay, perfect. Thanks. Okay, so as you guys know, we've been interacting with the King Abdulaziz Medical Research Center, KAIMRC, KMR as it's called locally. Now, our submission has been forwarded. It's been submitted to the IRB portal as of yesterday morning. And inshallah, as soon as we get the positive response from them and the approval, then the research collaboration would start with them. So this is a retrospective study. I'll share more information about it with you guys as well. This retrospective study basically involves them sharing Saudi patient data with us, and then we will retrain and validate our AI models on it. So it's a very, very good step in the positive direction. They're very excited about it. In the future, I will be filling you in about developments over there. For those of you interested, we can also share the content with you once we get their data and once we start retraining our AI models with them. So I think it's going to be good, inshallah. So it's a very major breakthrough over there. Hold on just a sec. Just give me one sec. Someone's messaging me. Just a second. Okay, so it was Nora from KSU, I think. Okay, so that's a very good positive breakthrough. Remember, the first time I visited their facility and met them was 2 months ago. So this was April the 20th or 19th or 20th. I went to them. So in 2 months, we've met them, we've done the submission, we've completed it, and we've submitted to the IRB board for approval. So this is a very, very fast track and expedited pathway. So that means they're very excited to partner with us, to collaborate with us from a research perspective, and they really like our solution. And you've got some serious heavy hitters over there, some very, very good research people, senior doctors. There was a doctor from Ministry of National Health Card as well. So that's very, very good. Inshallah, it's going to be better in the future. As soon as we start getting the data, then we can move forward with it. And so this is one thing. With the King Faisal Specialist Hospital, it's still in process. Let's see what happens over there. I don't know. Maybe things have slowed down during the summer. That's what I think. But let's see what goes on over there. So that's another thing. Aside of that, obviously, we signed the MOU with King Saud University, as you guys know, and some people join the training sessions from their side as well. And obviously, they have the AI lab, neuroimaging lab over there, which they utilize. During the week, we get inquiries from them as well, so we take care of them individually too. With Al Marifa University, the MOU still has not gone through. Uh, it's in process with their side, which is okay. We've also pitched a AI lab to them that we want to set up an AI lab at the university. So let's see how that goes. Maybe on my next trip, we'll see some developments over there. I will keep you guys updated on what's going on, and there are other things going on as well. So similar sort of a process is in ongoing with Princess Noura University, which is a very, very big university as well. And there are other organizations that we are targeting as well. During this week, I had a call with someone from Salama Hospital. Lady over there was very excited about our solution as well and how we can sort of collaborate. So yeah, things are going well. From a training perspective, this is the eighth session. So bear in mind that we have nearly 4 sessions to go to till the completion of the 12 sessions, which will be the first cohort. So inshallah, sometime in August maybe. I will try and come to Riyadh, and then we can have a small ceremony at Al Marifa University where we can give out the certificates to all the people who are participating, who come on the calls, like Haifa, Janan, Khulud, Maria, there's other people, people who submit their assignments and who do well and who participate. So they'll be getting certificates. And then we'll have a picture session as well. We'll put them up on LinkedIn. The ministry will be tagged and everything. I'll try and see if they can come and participate as well. People from the RDIA. We'll do a group photo and try and get the RDIA to put up a post for us as well. RDI is actually putting up a post for us this month too. It's gone for approval, so let's see once the approval comes. I'll share it with you guys as well. So yeah, this is what's going on. The plan, the mission for my next trip is number one, to give certificates to you guys, and number two, Mr. Azam is working on our portal, so I want to go to the hospitals in Riyadh and give them a demonstration about how our processes work and how our AI model works and how it gives you the diagnostic reports. So yeah, that's pretty much it from my side. I'll move it on to Mr. Azam so you can start the session today. Mr. Azam, please go ahead with the presentation. Thank you.
Okay.
Oh, and once the presentation is over, then we'll discuss the research task with you guys. High five, we'll have a chat with you on that. And Tartil, I hope I'm pronouncing your name correctly. I'll fill you in. I think it's your first session with us, so I'll fill you in about the research task and how you can go about attempting it. Meanwhile, Mr. Azam, please go ahead. Thank you.
Um, Wait a minute. Okay. Today I am teaching about something regarding the segmentation because there are a lot of models uh, which will provide you the explanation AI through the segmentation task. And today the idea is that how we explain in brief way the segmentation of 2D and 3D medical MRI. And basically this is a small tutorial, and also we share you some comparison about the segmentations frameworks. Basically the top frameworks which are using for the segmentation is free server. Many students who are working on medical imaging at some time of other 3D imaging regarding the segmentation. They already used the free server. The problem in free server is that the free server taking too much time for the segmentation of MRI image, especially when we give the MRI of vein, nifty and DICOM former, it take approximately 3 to 4 hours for single subject. And if you make a model and you give the more than subjects, at least 30 to 50 subjects for the comparison point of view, it's take a lot of time. Um, if you are using our NCI machine, it's take approximately 30 minutes for one subject. It's about 7 to 8 times faster to your normal PC. And now I also explore some other libraries, which is called fast server. It's take all the segmentation tasks which form the free server, but it takes too much reduced time. It take approximately 20 to 30 minutes for a single machine. And I also take experiment for our machine. How much time? My idea is that it take approximately 2 to 3 minutes for one subject. If I complete such experiment, I also share you the next lecture how much time it takes for approximately for the rendering of 50, at least 50 subjects for normal machine, and similarly to our NCAI machine, because it's great achievement if we are using such server for the production point of view. And the production, if the MRI, yeah, fMRI is segmented in 2 to 3 minutes. It's a lot of good achievement. And recently, such library is also introduced, and the paperwork is also launched in April 2026. And lot of people are shifting from free server to fast server. Now, first of all, I give you the brief idea about which is segmentation and how we done the segmentation in deep learning, especially the MRI segmentation. Um, this tutorial is basically based on the segmentation of all type of MRIs regarding the lungs, brains, and other things. But the idea is the same. How the segmentation is worked on. First of all, they gave some example of the NASA, some historical things. You said that the key point is that first is uh, how to this image segmentation come to the to the segmentation already is done for many past years. As you see that basically the segmentation is that this is three examples. First example is that the original example of a X-ray of the lungs, and you see that the area we spotlight there. Basically, this is a ground tooth. Ground tooth means the disease residing in such area. You see that the area of the lung is showing the black. It means some infection is in such area, and the ground tooth is actually infection which is detected by the naked eye, and the you see the image, and for the segmentation is performed automatically through some algorithm, and you see that the approximately 90 to 95% the area is the same, which is original ground truth. Basically, this is the idea of the segmentation, and you put such idea with the help of explanation AI methods, applying on your original image, especially in perspective of the Alzheimer. And we are also developing a app. Inshallah, 2 or 3 weeks, the app is live on, and I also demonstrate you different type of segmentations and gradient cam methods are applied on real time MRIs. Uh, Now I move forward that. Similarly, the some other example. This is example of the skin. You see that this is picture of the skin, and the some disease and infection area is added that. And after we are putting the segmentation prediction mask, you see that this is the original area, and segmentation mask is also exacted such area. Basically, this is the idea of the segmentation. And other example is mammography. You also apply the but the mammography is not our perspective at all. We are dealing especially with X-rays and MRI image, and we are just putting example of such.
Sorry, Azam, to cut you. I have actually a small task that we can assign to the students. So what we can do is, Azam, if you can over the weekend pick out one software. So it could be
Mm.
FreeSurfer, or it could be MATLAB, or anything that you find is good for a beginner level user. And you share the link in the group so that the students can download it on their computers. Ideally, it should not be a very big software so that everyone can download it easily.
Yeah,
And we give
today,
them a few MRIs so that they can practice on it.
yeah,
They can
yeah.
practice segmentation techniques. I think this would be a good task, good practical task for them.
Yeah, I suggest your people the software, which the software name is 3D Slicer. I also share you some tutorial in such presentation, and I also shared you the link where the software is available. This is very good software and easily used for such software. After completion of the such tutorial, I move to such tutorial. Now I move forward that basically the 2D also segmentation models. Basically the top of the line measure models which are belongs to the UNet. Basically, the UNet is type of model which are dealing to the segmentation. And recently, the Swin UNet is also a very good model which is utilizing for the MRI perspectives for developing the segmentation technique automatically. Now I move forward. Basically, these are the examples of the segmentation. You see that first example of images belongs to the ground truth. Ground truth means the original segmentation area, and the different type of models also implanting on such ground truth. First is that your swing U model, and after that your trans unit and attention unit and the simple unit, and the color is also shown that the area which is segmented. In our case, we are also segmented the area of the brain, which is also called the region of interest, RIO. and we also give you some examples and also give you tutorial how you apply that and how you, first of all, the idea is that you just go to such software 3D segmentation and also label that such areas, but it's give you the idea of the in the real time. You segment that manually the image for the doctor point of view, but if you want to go to some other ideas, you also use the fast server library. It's also automatically segmented the area. Now I move forward fastly. Basically, these are the two other good model bus, but these model are used the people who are program make a program या models for the segmentation. The first is the SAM. Basically, the SAM represent that medical segmentation anything, segmentation any model. Basically, the SAM represent the area, and the SAM model is very good model, is also trained that the medical images, and the medical example is MedSam. You also go to the MedSam. MedSam is also trained on the basis of the SAM model, segmentation anything, and it's very good model for the medical segmentation. And after that, we also apply the basically, this is the idea of that. This is basically a complete digestive system diagram. You see that, you see the labeling of the different segmentation area, 3D areas like the liver, rota, and other things. Now I am move forward. Basically, this is the medical SAM model. You see that we have the original your lungs picture, and you see that there is an infection of such area. The such area show that you see that the area is also segmented with the help of the medical SAM model. Now I move forward that we have another CT slice. CT means commutative demography scan, and you see that the slides by slide moves that, and segmentation areas also move that. You see that this is basically a good idea, a good example, which is utilizing by MedSam. And after that, we have some other good examples. We move forward. I also shared the link. There is also a coding link which is available if some people are interested for the medical scan segmentation. They use such examples. This example basically map on the 3D MRI. The MRI is a brain MRI, and you see that when the MRI is moved forward, and the segmentation area is also shrinking according to the line of the your slice number. This is very good idea and very demonstrative idea for you showing you the SAM model. Now I move forward. Basically, this is another good example of the other library. This is total segmented data library, which also you also segmented that the whole body of through such library. Now, basically, these are also the examples through the total segmenter. Basically, today I have not the idea to give you the practical demonstration for specific library. I am giving you some idea about how utilize such things. Now I move forward to the segmentation, top of the art free software for segmentation and other perspectives of the demonstration of the MRIs. Recently, I also give you some tutorial of MR Gross EL. Basically, this is also a 3D viewer software for the medical imaging, and in such software you also utilize that the you also utilize you also utilize that the such software for the 3D demonstration and the segmentations of the image of 3D images. You see that this is 3D slicer software. In the next lecture, I also give you the practical demonstrations. Today, I am also give you some idea, because in single lecture it's not possible for me to give you the practical example and also the demonstration. The idea is that you give you the high level abstract in one lecture and covered in the second lecture topic through some practical demonstration example. You see that the 3D software and simply in real time we have when we take MRI from the scanner put the images in the form of DICOM format, and the DICOM format residing to the patient, especially the patient ID, the ID allotted by some hospital, and all the images are goes to such folder. You go to the 3D slicer and simply add the DICOM data. You see that in software you have import DICOM file. When you select that, the folder is selected that, and all the files are resided in the pan. You see that there are different DICOM format files are resided in the pan. Now we move forward that the all the files are available you in the form of such a way. Basically this is example of some lungs type image, and you see that the DICOM file extension is dc called DICOM dyn. Basically these are the extensions of DICOM file. And after you see that there is now the file is loaded in the pan of the 3D slicer. Now we move forward to the segmentation. Uh, Wait a minute. Uh, Now we put the segmentation of 3D slices. You see that we have an idea. We put the image of the some your image of, I think it's an image of your belongs to your lungs type MRI, and you see that there is some fat tissue, some glandular tissue, some muscular tissue, some lung tissues are available. And the idea is that after such image, and we segmented all the areas which is labeled at. First of all, we label the fat tissue, second the glandular tissue, third the muscle tissue and lung tissue, and last the space air of the body. Now you see that we add a button, and after that we also source some it's just like your uh, some atlas on that. Atlas means vapor on that. And after that you see that when we put a atlas, now we selected that the colors on that. You see that the atlas is mapped at, and after that these are the colors which shows the such thing. First color is shown to fat tissue, muscles, lungs, and other things. Similarly, we also put some um, manually some buttons. In next lecture, I also explain you how the segmented that. Basically, the idea is that how you puts and work on that. And lastly, these things are also wholly segmented. You also export the 3D mesh in the segmentation form. Now, if somebody have some question regarding that, then we move forward.
So, Mr. Azam, I have a question. So, these guys can download the software, right? Or
Yeah,
they
yeah.
have.
First, I am also giving the software link. The software link is slicer.org. Basically, this is a 3D
Okay.
slicer.
Okay.
I also share that in the chat.
Yeah,
Uh,
yeah. Please share it in the chat so they can download
uh,
it. And what
yeah,
about
yeah.
the sample MRIs that they have to practice on? Will you give them to the
Yeah,
students?
yeah. I also upload the sample MRIs for regarding to the brain
Okay.
in the our repo. No,
Okay.
neurocognitive or repo is also upload that. I
Okay.
also give you
Perfect.
the both the tutorial links. First tutorial link is that, and the second tutorial link is also.
Guys, please note these down so that you have the relevant links. And yeah, just play with it. Just download the software, use some MRIs, and see how you find it. It's just a matter of playing with it. The more you play with it, it's like a video game. The more you play with it, the better you'll get at it. So inshallah. Back to you, Mr. Azam. Thank you.
Okay, now the second task is that first of all, I give you the idea about the segmentation. Basically, the segmentation idea is too much hot. When you point out, you localize some MRI positions of the disease, you highlight the tumor, you highlight the tissues and other things. You also have some idea of the segmentation. Basically, these library and these things are which are also demonstrating you. You manually segmented that with the help of such software. But now we move forward that how uh, you automatically, how you with the help of some software, you have the help of some atlases. Basically, the things are just like your tracer, uh, images of MRI images. You just put your images in the frame and also apply the tracer. The tracer also highlight that such areas, and you put some gradient cam and other things on the tracers. Now I am moving forward to the fast server. Basically, um, there is common free server is also a utility which people are using for the segmentation of different type of brain images and other things. But in the brains, you especially apply the free server. The neurology, like brain tumor, like Alzheimer, Parkinson's, and other diseases, we are using the free server for the segmentations of the different areas. Now, but the problem is that the problem is that the segmentation through the fast server is too much slow, and it needs lot of resources. When we put single subjects, I have a machine of Core i7 machine, and I have approximately 32 GB RAM and Core 13 generation laptop, and I practically apply that. It take approximately 3 hours if you have a good processing power plus internet access. All the things are matter that, and after that 3 hours you just put experiment of just one single MRI. In case there is some problem, some other cheese, your machine is also heat up. If you apply the long run of things, the all things are matter that. But in case of fast server, I also apply some little experiments on that. It takes approximately 30 minutes, 30 to 35 minutes, depends upon your machine, because my machine is good, and it's take approximately 30 to 35 minutes uh, in applying the fast server. But the results are totally same. Approximately 90 to 95% the cases are bill approximately same as compared to the free server. You see that, and the commands are already same. I also deeply experiment that how the things are managed. That the things which are done 3 hours and the things are done in the 30 minutes, and the accuracy and the demonstration is also a similar. You see that, also you see that basically they use some looping and some other ideas. You see that if you apply the service recon, you see that it takes 1 hour in fast free server. But in case of the fast server CNN, it takes 1 minute. You imagine that one thing is done 1 hour and one thing is done 1 minute. It's a great idea. Now you see that basically I show you the official documentation of the server. Basically, the Deep Medical Imaging Lab is a innovator of the research library, and the research paper is also available. I also share you the research paper. First, you just read the research paper, which things are good, which is are and compare that. Basically, the idea is the free server map the tissues. You see that the different type of tissues are segmented through the fast server, and also that it's calculate the subcortical and your corticals. Basically the corticals is are the such tissues areas which are segmented that, and after you see that there is a volumetric segmentation. Volumetric segmentation mean we give a volume, and it volume have a different slices, and after you put the slices, the framework utilizing the UNet and some convolutional neural network, and after that you see that different basically this is a raw library, and is apply the
Mr. Azam, are you there? I think we lost you. Could be a power issue. Sometimes you get these power surges. Is everyone else there?
network architecture segmented all these three type of views. And now we move forward that you see that there is a iteration of the segmentation, how the iteration process is in case of free server, and how the iteration process basically this is the iteration process of the free server. It take too much time, and this is the iteration process of the fast server. It take one shot mapping, and it take iteration inflation. Basically this is the idea which is the difference between the pre-processing, because in case of free server you apply different looping ideas, but in case of fast server you just give one shot mapping. Basically this is a mechanism of fast evaluation of fast server and the free server. Now this is a proof of concept and other things. Basically this is a corneal effect. Basically this is the comparison of both the image. You see that if you take after 3.5 hours, and you give the images of the corneal effects like free server, and you see that after 30 minutes the similar that the images are approximately similar type of thing, but the time difference is too much. One thing is takes time more than one thing is 7 times shorter. Basically this is the idea which we are utilizing in the professional point of view. Now this is the paper uh, of the fast server. I also share you the paper, you read that. Basically this is a narrow imaging pipeline, how this library is a framework is worked at. Basically you read such paper. It's a good idea first of all which framework you use. First you use their paper, your research, how the people are working on that. You see that first of all I'll give you the idea of the 3D architecture. Similarly, the giving you the idea of the whole brain segmentation, and use which we are utilizing that. You see that the fast server, the moving of the fast server, how the raw images converted to the CNN architecture, and after applying that, the surface recon. Basically, this is the 3D segmentation. This is the normal segmentation, and after surface recon is applied, you see that all the things are in 3D format. These are 3D formats, and after that, uh, they are also applying that the methodology, and after you see that there is a pipeline things, and these are the accuracy and comparison. Basically this is the comparison how the things are managed for different type of data sets. They take data sets of acne, OSIS, and other some good organizations. Now you see that basically this is a surface dice which are created through the fast server, and after that you see that there are different type of comparison. This is another comparison. You see that In other comparison, the things are comparing. One thing is doing from free server, and you see that similar type of things are done in the fast server. And after that, this is other comparison. You read this paper, it's came approximately 2 or 3 days, but there are very good things, especially when you read such things. Such thing is that which type of structures are labeled for segmentation areas. You see that your cerebral white matter, your lateral ventricles, these are all the terms. Your hippocampus, these are all the terms which are you are familiar regarding the brain prospectus. There are brain anatomical structures. There are the subcortical structures, and these are the cortical structures. And this is very interesting. You also take a MRI and label that the all the structure through the fast server, and this is also a very good research. And I also apply such thing when we are doing on the KSHF lab, King Abdullah Research Lab, and I also applying such things because these things are very state of the art. And you see that the library introduced in 2026 as latest status of the art library, and you see that the paper the publishing date of the paper is I think April 2026. There is some date also mentioned that. Okay, 2020 the paper, but after the recent paper is also available. Now we move forward. Also show you the also share you the fast server free examples. You also move to these such like that. If you go to the free server comparison library, you see that I also shared you the free server comparison library. Basically, this is a comparison. Somebody work on that, work on free server and fast server also. You also check such library for the demonstration point of view. And the other library, which is the original library, which is library of the free server, fast server, sorry, the fast server library. The library also overview that I also share you the fast server library. Wait a minute. You also go to the library and also see that how you install that. The library basically, if we move for the get started, you see that there is also example of 3 ways to install the free server. Basically, I suggest you are using on Linux machine, but the result of the Linux machine is too much cut. And also you use using the Docker environment, because the Docker environment is also too much good for the rendering of the service. It's move on. You see that we have the examples of the Docker library, and we have also example of quick segmentation. First of all, I move to the quick segmentation. You see that the quick segmentation, you just install the free server. How the free server install? First of all, this is a basic question. How I install the free server? Simply, you go to the tutorial folder, and you see that in the tutorial folder you have some installation. First of all, you have first installation is you have is the Docker installation. First of all, recommended is you install Docker. If you have no idea installing the Docker, it's not worried about that. You go to the local installation. In local installation, you just use the Python, and Python install in your side. If you are no Python, you install the Python through the command, and after that you install some setup tools command, and after that you apply some some such command, some other libraries, and after you install such the Docker framework. Basically, the Docker installation, how to install the Docker. It's the other long lecture, but I am not moving to the Docker installation. And if somebody have a problem for installation, now I give you some tutorial about the installation point of view. Now I move to, first of all, the first notebook, quick and easy free server installer segmentation in 3 clicks. When you click on that, you move to the search tutorial library. First of all, this is the Google Colab. If you have not a machine, you just log into the Google Colab. Google Colab is a free for the rendering of the fee server. You see that tutorial Jupyter notebook file is available on that. And after that, you see that we have first of all the fast server setup. You just go to the that and run that. But one thing is that you have Google Colab. You have your account on Google Colab, and you have some settings that the setting is also available. You also create the a folder which name is fast free server tutorial. Basically, this is a library which are normally we are available. The library is content. You also create in content the sub library, which library is named at fast server tutorial. Now I am moving forward fast and forward in next lecture. If somebody, first of all, it's your assignment, you run at your side. If you suffer some problem, then I in next lecture I help you how to install the things. Now we move forward that we just uh, get the free server from your original repo. Basically, GitHub DB is a path of the repo, and after that, you start the free server installing, and after installing the specific library which are needing from the free server, we need the PyTorch, we need the Yard, Plotty, and after that, all the things are done. You see that such like of message. Today I am not applying because it takes some time. All the libraries are tying that I also compile these things and just show
Yes,
you.
I don't think that that is necessary. But I think the students can just download the software that you recommended, and then they can use the MRIs and just play around with it. So this last 4 weeks that we have now, there's two things I want to focus on. Number one, I want to get the students to do something practical. So practical could be, of course, they download the software and then they get some MRIs which we give them, and then they start practicing on it. Mr. Azam, please also share the links in the WhatsApp group. Number two, what I want everyone to do, I've also messaged in the group is that whoever is on working on the research task and they pick out a paper they like and then they study it, I want them to make a either a brief analysis or a brief presentation, and I want them to speak on this training session call and tell us about the paper that you have read, what you have learned about it. You can speak maybe 5 to 10 minutes on it, so that it's for the benefit of everyone else. If like 5 or 10 different people read different papers and they tell each other, everyone about it, that's something new in terms of knowledge that then you are sharing with everyone. So I think that is very, very important for everyone to do. Hi-Fi, I know you took an attempt at the research task and you made that wonderful table. We saw it and we actually shared it as a template, as an example for the others as well. So pick out a good paper over there and work on it. And then maybe in the next couple of weeks or so, you can speak about it. I want everyone to speak about what they're researching. So that's very important. And Mr. Azam, I think that's it for today. It's a shorter session. I know it's the summer holidays. People are on leave. A lot of people messaged me. Khulud just messaged me. She's also has guests over at her place. So it's just that time of the year. But it's okay. I admire, respect the people who are showing up, especially Haifa, Nora. Thank you, Tartil, for showing up today. I think it's your first session. Tartle, would you like to tell us a little bit about yourself, what you're studying, where you go to school? I think you go to Al Marifa, but what are you studying and what are your goals? Maybe 2 or 3 minutes if you'd like to speak.
Yes, hello. First of all, I am Tertil Marwa. and I am a medical student in Al Marifa University. I am at 8 level now, and I finished the basic levels. Now I will go to the clinical levels. Also, yes, my goal to be to take a lot of courses in AI, not especially in neurological, but also in any courses in AI also. Yeah, that's it. And yeah, that's it. Thank you for all.
Perfect, perfect. Thank you very much for that short introduction and welcome to the group. Yeah, I understand that you are interested in AI. It doesn't have to be restricted to neuroscience. We can also share other stuff with you, which you can then use in other applications of medical sciences, whichever your area of expertise may be. So I think it's a very good platform for you to join. What I will do is I will share our research task with you as well. Although you've joined in, we're barely more than halfway through our training cohort, but that's okay. You can also join in on the next one, which will begin, inshallah, soon, right after this one finishes early August. And I'll share the research task with you as well, and then you can start working on it. You can share some notes with your friends and colleagues that you make in this group. And everyone, I'd like you to post in the group as well. Post articles on neuroscience and AI and whatever you're researching. I just want it to be a forum where everyone can interact with each other. You've got all those people from Al Marifa University in the group, so I want you to make new friends over there, new colleagues amongst yourselves, and yeah, that's about it. So till next week. Next week we'll give you the agenda of the training session early next week. Meanwhile, you guys, you go and you download the software, get a couple of MRIs from the GitHub repository that Mr. Adam shared, and just play with it a little bit. See how you feel about it. It's very interesting. It's something new that you learn. And Mr. Adam, if that's it, then that's the end of the session. Unless you have something to add.
No, it's okay. The links are also shared in the chat. The documentation link of 3D slicer is also shared in the slide, and software link is also available in that. I also upload the MRI and regarding to different labeling like Alzheimer's disease, mild cognitive Alzheimer's disease, and the patients who are healthy for different data sets. Some more like the 4 or 5 subjects, 2 or 3 ID and 1 or 2 3 on the OCs. And it's good if the people are segmented the areas. It's no new world and new experience for the beginners, and I thought it's taken a lot of fun for such people.
Haha, perfect, perfect. Inshallah. So thank you very much, everyone. If you have any questions, you can always message me, Mr. Azam. You can message in the group as well. And thank you for Nora as well. Nora joined in. Do you have any questions right now, Nora Haifa Tartel?
Yes, please. I want to ask you, can I search with you in the research and share with you, or in this research, of course, or what?
Sorry, how do you mean? Please come again.
Can I share with you in the research in the current research now?
Yeah, you can share. Are you saying you've already done research that you want to share? Is that what you're saying?
Yeah, I have some research. I will search, but can I put my name also with you in this research?
Yeah, you can. So we are publishing a paper with King Abdulaziz Medical Research Center. So over there, you will not be able to put your name.
Okay.
But with the research that we're doing over here,
Yeah.
obviously, yeah, you can use your name, you can attach it to us, and if you'd like to publish a paper, we can help you with that as well. So that is not a problem. And
Yeah.
whatever so the research that you've done, is it in neuroscience or AI? Which subject have you done research in?
No, I had finished a research about diabetes,
Okay.
but not in AI. Yeah.
Not an AI. Okay. Okay. Well, yeah, you're welcome to share it with me. I'd be happy to take a look at it. But the research task that we have given out is in neuroscience. So I would suggest take a look at that as well. It's not very difficult. You have to go through, find a few papers you can study, and then you can learn about it a little bit. I think it'll be good to broaden your horizons a little. So I'll forward you the research task that we've given out earlier.
Tretal, are you have some idea about the Python programming and some PyTorch and CNN, such like models? You done that in past?
No, actually, no.
No, she's Mr.
Okay.
Azam. She's a medical researcher and medical
Basically we are doing the research on such like things, and if you have not idea if you how you manage the research evaluation metrics and some other experimental work, because our work is deeply involved in such like things like modeling and some explanation AI and other things.
Tatul, what's the research? What's your perspective on the research that you've done for diabetes? How do you do that?
Yeah, it's about the awareness of type 1 diabetes
Hmm.
among the broad population.
Hmm.
And yeah, we finished cross sectional research. We
Hmm.
go to molds and make a randomly sized uh, s- uh, we take
Total,
a random size
are
population.
you Turtle, you have some idea about the ranography on diabetics datasets? Because the ranography is a AI technique, how you take the datasets and apply the ranography on the diabetic type 2 datasets and evaluate that the person diabetes condition. Are you
Mm.
work on that?
Actually, no, I don't have anything in imaging in AI. Uh,
Uh,
yeah.
I, I
Basically,
I think what you're trying to say is that you've done research on the population segment in Saudi Arabia. Is that correct? That you've done questionnaires
Yes.
and gathered information on population and the segment of the population and the age brackets that has diabetes and so on, right?
Yes. Yes. That's it. Yeah.
Do you
but
want to
you
do something? Sorry, do you want to do something similar for Alzheimer's as well? Because we also have a research group that we are going to start using to collect data on Alzheimer's and do something similar that you've done for diabetes, but do it for Alzheimer's. Would you be interested?
Sir Ahmed, one idea is
Yeah.
that
Yeah.
if he has she has a sufficient knowledge on diabetic, then
Mhm.
they move to the ranography.
Mhm.
Ranography basically the idea you got the images of the diabetic person, like the diabetic person have some foot infection, some infection on the hands, and some infection on the put on their body. They
Mhm.
take some infection images basic and apply some similar model like segmentation models and some CNN and the things. Built-in models are also available. You
Mhm.
not train the models, you just put the models which are also available, and you done very good research evaluation metrics because she has sufficient idea about the diabetics and the basic knowledge and other things. And now
Mhm.
just they put the wrapper of the AI, and Google is also providing online the ranography features of the diabetics. Recently, such topic is also very good topic for AI perspective.
That's good. So, Tardal, how does that sound? What would you like to do? Does that interest you?
Yeah, that's interesting. So can you but summarize the idea, please?
Yeah.
Okay, so what Mr. Azam will do is next week, Mr. Azam, if you can share one or two links with her where she can
Okay.
go
Yeah.
and download that software, which is going to help her with that process that you mentioned. And
Okay. Okay. Fine.
Okay, okay, sounds good. Sounds good. Anyways, guys, thanks for joining in, Mr. Azam, over the weekend. If you can please share the training session video link and the transcript so that everyone can benefit from it. The people, especially who were not able to make it to the call. I see Hasib has come as well. Hasib is a little bit late, so we'll mark him late today. But it's okay. Thanks for joining in. And I'll see you guys inshallah next week then. And stay tuned. Remember, please join our LinkedIn page, like our posts, spread with your colleagues and friends. And see you soon, inshallah. Thank you very much.
Okay, Allah.
Khudafis.